Hello, everyone. This is Kelly Eversole. I'm the Executive Director of the International Alliance for Phytobiomes Research, and I'm joined today by Paul Cotter from Teagasc in Ireland. So I'm pleased to have him here as part of our webinar series. We'll wait just a few minutes before we get underway to make sure everyone has a chance to join us. All right. So I want to welcome everyone to our webinar series. This is a core group of webinars that we've been providing since last fall to kind of highlight some of the activities going on internationally. And as I mentioned previously, we're highlighting today an EU project. And but before we do that, I would like to tell you a little bit about the International Alliance for Phytobiomes Research. We are a nonprofit consortium of industry, academic, and governmental scientists. And this webinar is made possible by our sponsors. We have a wide range of sponsors from various startup institutes, as well as international or governmental organizations. So before we get into the webinar, I'd like to just kind of have a quick explanation of what we mean by phytobiomes. Phytobiomes really is used to describe the complex system of plant-based agriculture. It is the idea of a plant in a specific biome or a specific environment. In this case, as in the picture, it's soils. And it includes all of the interactions, both the biological, such as the microbes or other animals and plants, as well as the geophysical, the soils, the weather, water, climate, etc. And all of these are influenced by management practices that a grower or farmer may actually use, or even a forester or a grassland manager may use. So the Phytobiomes Alliance vision is the idea that by 2050, all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for a specific field in any given year. Our next webinar after this one will be on the 9th of July, and we'll be looking or hearing from about an exciting project that has just been published in Science related to genomic approaches to the epidemiology and diagnostics of phytopathogens, tracking the global spread of agrobacteria. You can register by going to this link, and these slides are actually available for you, and it'll be at the same time as we've had it today. So just so you know, the webinar is recorded and will be posted on the Phytobiomes Alliance YouTube channel, usually within a couple of days, sometimes as early as a few hours. It just depends on how long it takes for the platform to actually prepare the file. The presentation section will be followed by a set of questions and answers. If you have any questions, please submit your questions in the Q&A panel. Do not use the chat, as that will not be, we will not be able to see that. You can use the chat panel to talk with fellow attendees or the organizers, and you can also download the handouts from the presentations right now in the handout panel. So without further ado, I'd like to turn it over to Paul Cotter, who will tell us about the EU Master Plan. So Paul is the co-coordinator or the coordinator of the EU Innovation Action Master, which stands for Microbiome Applications for Sustainable Food Systems Through Technologies and Enterprise. So thank you for your participation today, and I'm going to turn it over to Paul.
Super. Okay, thank you very much for that, Kelly, and thanks indeed for the invitation. It's really appreciate the opportunity to tell you a bit about our project. And while I've presented on it briefly in the past, this is probably the most comprehensive presentation I've given to date on it. So I've pulled together a lot of information for this, and hopefully I do it justice. I'm conscious that there is time at the end of the presentation for questions and answers, but I'm also happy to answer any questions subsequently. Either you might want to direct them to me by email or via my Twitter account, which is listed there. And I'll also direct you to the @master Twitter feed and also our corresponding website too. Okay, so why study microbiomes and why have the EU invested in this so heavily? And so I think there's an enhanced appreciation, as is the case in the US, of the importance of microbial communities and how they contribute right throughout the entire food chain, from the marine to the soil, plants, animals, and consumers, waste streams, and all sorts of other activities. The challenge, though, that we were set when the EU decided that they should fund research in this area relating to the food chain is that they asked that we submit a project that addressed all of these different niches in parallel, which was quite a challenge, but is one that I think we're moving towards meeting, and has been particularly exciting working with lots of different people from throughout Europe and with others elsewhere, including the likes of Kelly, who's part of our advisory board, to give us guidance as to how best to proceed. So I'm conscious that most of the people who'll be attending here today will have some knowledge of the microbiome and the sort of revolution that has taken place in the microbiome research in recent years. So I won't linger too long on that, but maybe just a slide or two for the benefit of the small percentage that are maybe not so familiar with that background. And so I guess it really relates to the evolution and the transformation in the field, whereby there's been a movement away from conventional microbiology and agar-based approaches and growing microbes in essence, in recognition of the fact that it's very difficult to grow all of the microbes from any one community at the same time, because you would require multiple different agar plates, lots of different temperatures and conditions, and then there are nearly always some microbes that are very challenging to grow in isolation anyway. They require other microbes. They may come from the gut and require anaerobic conditions, and ultimately we know in certain environments that we really only just see the tip of the iceberg when we use agar-based approaches. Fortunately, as a consequence of technological advances relating to DNA sequencing approaches, it's now removed our need on culture-based strategies, and instead from any community, whether that's from an Antarctic region or a soil or a volcano or the human gut or a termite's surface or leg, then we can look to see what microbes are there by virtue of first extracting the DNA from it and then studying to see what's present on the back of that by carrying out bioinformatic analysis. And so I may refer as we go along to metagenomic DNA. And what I mean there is that I refer to all of the DNA from all of the genomes from all of the microbes from a particular microbial community that we are studying. And so in the past, there was a big focus on, after extracting the DNA, using approaches like 16S amplicon sequencing to determine which bacteria are present in a community, or to use ITS sequencing to determine which fungal communities are present. But increasingly, there has been a move now towards shotgun sequencing, which wasn't used previously because of associated costs. But as the costs have improved and the corresponding bioinformatic technologies have improved, now this is rapidly becoming the go-to approach. And the beauty of this sort of approach is that it enables one to not only look at the community that's present in terms of either the bacteria or fungi, but in fact, you can look at both of them simultaneously while also looking at phage, archaea, and in essence, every microbe that's present in that community together. And that's because instead of just focusing on one specific barcode gene to determine the taxonomy, instead you can sequence a cross-section of all of the microbial DNA that's present, hence the term shotgun sequencing. And through this approach and the comparison with different databases, one can get down to a species level assignment, which isn't typically possible for amplicon sequencing, and, for example, bacteria. And indeed, one can get to strain level analysis, especially if you assemble that DNA sequence information into MAGs, or metagenome-associated assembled genomes. And the other beauty of the approach is, in addition to being able to identify the bacteria and yeast and so on that are present, one can also look to see what metabolic pathways are there. So, for example, within a particular food, one can determine if there are lots of genes in there associated with healthy aspects and the production of, say, for example, short-chain fatty acids in the gut, or are there lots of genes associated with toxins and antimicrobial resistance, in which case you then know that there's a risk associated with that food. And so it really is a powerful technology. And if you take that further and combine it with other omics approaches like metabolomics and metatranscriptomics and so on and so forth, you can really get a very deep insight as to what's happening within a particular community. So this ambitious master project, well, we would not have applied for it or assembled a consortium if we weren't already to some degree active in this particular area. And so due to funding from the Irish government and from, for example, APC Microbiome Ireland, in which I'm a participant, it was possible to already ground our research in this specific area and develop some pilot data that allowed us to then leverage with a view to putting this consortium and applying to the EU for a larger amount of funding. In our particular lab, this background related to looking at initially at human microbiomes and of the gut and fecal material and of animals. But then in maybe 5 or 6 years ago, we began to increasingly employ these new microbiome approaches to other microbial communities, including fermented foods and food processing and food production environments. So over the years, we've applied this to things like water kefir, kombucha, kefir, which is a fermented milk, kombucha is a fermented tea, in order to determine which microbes are present. Because there's been such a rebound and an increased interest in fermented foods in recent years, then there was a great attraction to add some more science to the claims that were being associated with these foods, to determine which of them indeed were most healthy and which of them had potentially health promoting microbes present therein. How do they contribute to the flavor of the product, and in some instances spoilage. And in one instance, the example I'm showing here relating to Nunu, one can also apply these technologies from a food safety perspective. So Nunu is a fermented yogurt-like milk product that's produced in Africa. It has a relatively short life, in that essentially what happens is that the Nunu, or the milk, is stored in a container, and it's allowed to spontaneously ferment, but also, I suppose you could say, that spontaneously go off. And so there's kind of a rapid competition between healthy microbes and potentially disease-causing microbes. And we were asked to help within a project where we were trained and untrained producers of Nunu, some who were trained in HACCP protocols and others not, provided Nunu, and we carried out a metagenomic analysis and compare the species level assignment across those. Ironically, it didn't quite turn out as we planned, in that those who underwent training had much higher levels of pathogens present in their Nunu, higher levels of Enterococcus faecium, Acinetobacter, E. coli, Klebsiella, and so on and so forth, which we ultimately attributed to the fact that the HACCP training resulted in a movement towards plastic containers rather than the original natural containers that probably had some natural fermenting good microbes present therein. And the trainers inadvertently moved the producers away from that. But it was interesting nonetheless to see what the consequences were. And we were able to determine, for example, that some of the E. coli that were present in the Nunu were closely related to some that had been responsible for outbreaks in other parts of the world previously. Now, this is very much an academic exercise, though, and so the question is, could this be applied in a manner whereby a food producer could very rapidly harness these technologies in the future? And I'll come back to that later. Another way in which one could apply it relates to the production of milk, and this was a study that was done in Ireland, and the production of raw milk in Ireland at different times. So in Ireland, we have a seasonal factor, so the cattle are outdoors for most of the year, but during the winter season, they are indoors for a brief period of time. And so this abstract art, I guess you could call it, does have some sense. So I'll talk you through what exactly you're looking at here. So in essence, we collected lots of samples during the indoor season and the outdoor season. So each dot here represents a microbial community that was generated through sequencing data. And so each of them represents thousands of individual sequence data points relating to the microbes that are present in a community, and these are coalesced. And so if you're not used to looking at these sort of PCA plots, what's the key point is that if two symbols are located close to one another, it means that the microbiome is quite similar. And if they're far apart, the microbiome is quite different. And so we collected the milk samples from the indoor and the outdoor season. These are the circles here and here. And then lots of other environmental samples from fecal material, from silage, from grass, and so on and so forth. And so if you look at it here with the different clusters, you can see that, for example, the milk and the teat surface of the cow have microbiomes that are somewhat similar. Indoor feces has some closer relatedness than, for example, silage during the indoor season. And during the outdoor season, you're seeing again milk samples here, outdoor feces being an important contributor, teat surface also important, but not quite so important, with grass and soil less so. And so this is starting to give us an insight as to different sources of contamination, what we need to do in order to increase the quality of the milk to ensure that particular microbes aren't transferred from these environments into our raw milk. So we're gradually moving more and more towards translation. And this is important when I come to master, in that master is funded through an innovation action, which requires that what we do needs to be very translatable and to be employed at a high TRL, high technology readiness level, and not just be blue skies type research. Now, another one of our pre-master projects that kind of set the scene related to our application of shotgun sequencing to try to determine an issue relating to a commercial problem that very many cheese producers have around the world. And this is a pink discoloration defect. It particularly affects Swiss, cheddar and Italian type cheeses around the world, and has been in existence and has been first reported since the early 1900s. So lots of researchers have put a lot of time into this without any great success. And we were asked to contribute to a project where a PhD student was coming towards the end of his studies and realized that he really hadn't much to show for them because he, like very many others, hadn't been able to figure out what the basis for this was, whether it was biochemistry, microbiology, and so on and so forth. So rather than using conventional microbiology, this PhD student gave us some samples from controlled cheeses which did not have the pink discoloration defect, and a number of defect samples from different suppliers. And you will see an immediate difference between the two in that while all of the cheese samples had traditional cheese type microbes such as lactobacillus, streptococcus, and propionate bacterium, those from the defect cheeses differed in that you see this extra lilac color here representing microbes from the genus Thermus, and most specifically Thermus thermophilus. And so this intrigued us, and when we read some more about Thermus thermophilus, and some of you might be aware of this already, that Thermus thermophilus is a carotenoid, in other words, a color-producing microbe that's generally found in places like Yellowstone National Park and other rich environments where you find hot water and high temperatures. And so high temperatures is an issue relating to the production of these type of cheeses. And when we try to determine and try to extend our experiments to really establish if Thermus was genuinely responsible for this phenomenon, what we did is we isolated some Thermus from these cheeses and then recreated a cheese by spiking back in this Thermus microbe, this bacterium, to see what happened. And we did indeed recreate it, kind of the equivalent of a Cox postulates for food, recreate this pink discoloration defect in that cheese, and therefore identify the microbial basis for this phenomenon. Now, this microbe isn't a disease-causing microbe. It purely causes a discoloration defect that results in these cheeses being removed from the market and causing considerable economic losses. And we went further and we looked at environmental samples from the pink production or the cheese production environment and found this Thermus to be particularly enriched in hot water sources, again, consistent with the places where you typically find Thermus microbacterium. And so that allowed us to develop, to identify sources and to develop strategies for industries to remove it from the food chain and therefore remove the problem in the longer term. And then just one final background. So continuing on with the cheese or the food production environment or dairy production environment, one other scenario, one other issue or study was one whereby we collected samples from a process involved in production of milk powders. And so this involved collecting samples from 67 different farms, from the bulk tank milk of 67 different farms. All of this milk was combined across 11 different tankers and then went into a whole milk silo and was pasteurized and went through all of these individual steps, evaporators and so on, until you ultimately produce some bags of skim milk powder. And we traced the microbial transfer right throughout that whole process at 2 different times during the year. So again, I mentioned you have an indoor and an outdoor season in Ireland. So we looked at mid-lactation during the middle of the year and also late lactation. Now, these were just 2 single days, 2 single time points. And so they don't represent those seasons in general, but they do give us an insight as to how the microbiome can change and how microbes can move through the chain differently at different times of the year. And so in particular, I'm going to highlight the last 2 sets of bars here and bring to your attention how they differ from one another. So at the start here, where you're looking at milk, in the bulk tanks, you see lots of similar microbes, lots of pinks. These are pseudomonads and other such cold-loving bacteria. And these pinks, these pseudomonads, continue to persist in the late lactation example here and other such related microbes. Instead, though, during the mid-lactation phase, we saw a large shift right towards the end of the food processing chain, whereby there was a change towards spore-forming bacteria. And these seem to have entered not through the milk at the beginning of the process, but rather have entered in the form of biofilms that were present within the plant that sloughed off and entered into the milk and caused contamination. And because of their thermostability, persisted after pasteurization and other high temperature treatments, and therefore were found in the milk at the end of the process. And obviously the risk then is that if you use those powders in other products later on, these spores can germinate and cause problems. And so by getting this sort of insight, we can extend that now to looking at over different times of the year, more time points, and also determine how different cleaning practices and so on can impact on which microbes succeed in getting through the chain and entering into the final product. So after that, the provision of that background, I think you will gain a sense as to why we thought it would be interesting to carry out these sort of studies on an even larger scale, and hence the project master, and hence this master plan that I'll refer to over the next number of slides, where I'll do my best to do justice for what really is a very large project. And I guess under other circumstances, it could be regarded as a daunting project because of the very many different tasks and areas that we are targeting, but it really hasn't been like that at all. It has been a real pleasure to work with some really fantastic scientists and companies right throughout the continent who are very much passionate about this and are also passionate about applying these technologies to really make a big difference to people's lives going forward. So these are all of those passionate researchers that we deal with. This is a picture from our most recent in-person meeting that took place in Italy, in Naples last year. We were due to meet in Madrid earlier this year, but we had to do that virtually instead. And you will see the geographic distribution of the various different partners, and we'll see that very many of them are universities, but then also some of them are very large multinationals, the likes of Anheuser, AB InBev, Qiagen, Christian Hansen, Danone, and others, who are really interested in the microbiome space and applying these technologies going forward. And again, this reflects the fact that we are very much obliged by the European Union that this research is not blue skies research, but will come up with new products and new strains and new guidance for industry and for consumers. So what are we doing? So we're taking a global approach to the development of microbiome products, and these will include foods and feeds, different services, or different processes with high commercial potential. And this is with a view to improving the quantity and quality and safety of foods, multiple food chains. And as I said, we were obliged to be very ambitious, and we're including aspects of marine, plant, soil, rumen, meat, brewing, fruit and vegetables, fermented foods, and ultimately human health. So these are all of those various different areas that I mentioned just now. But in addition, the various different specific product areas relate to looking at different strains and consortia, looking at services and processes, new feeds, foods and feeds meaning for animals rather than for human consumption, different new approaches to molecular analyses, and also to generate new databases and improve pipelines. And that while microbiome research has moved on for certain niches, for example, the human gut, it's somewhat ironic that for very many parts of the food chain, even though lots of the microbes are culturable, the databases and the number of genomes and metagenomic databases or sequences that are available are much fewer. And therefore, if one wanted to apply this technology in industry right now, it's not so practical because there are lots of reads that just are not easily assigned to microbes that are in the food chain as a consequence of these imperfect databases. And so we're trying to contribute to providing a solution to this as well. So getting into a little bit more detail as to our specific objectives. And so this relates to, in the first instance, improving important traits for fodder crops and to employ microbiome diagnostics. I'll return to all of these in a few moments in greater detail. Improving bioavailability of sustainable feeds for the marine industry, and to also to assess fish health and to detect pathogens. From the rumen perspective, we want to increase feed efficiency. In other words, that means when an animal consumes feed, that is converted into milk or to good quality meat, rather than being lost to things like methane production and urea. We want to further optimize fermented food microbiomes and also use fermented food microbes to improve the value of waste streams. So materials that at the moment would have to go would be waste and are an inconvenience to the industry, to instead add value to them by fermenting them and adding new metabolites to the products that are of value within foods and feeds. And then looking at how all of these different new products impact on gut microbiomes in animals and in humans. We also, as I mentioned, are improving different software tools and new reference databases that will not only be used for master, but will also be used for the whole community at large. And so we don't want to be just simply insular in this regard. We want to bring the whole food industry along with us. And importantly, to develop a knowledge exchange plan so that we can make sure that the information that we generate is dispersed well to consumers and to the food industry. Okay, so as you may have noted, I'm gradually getting into more and more detail as we move along, but this will probably be as detailed as I get for some of the specific niches, simply because with the time frame available, it's not possible to cover them all in very great depth and to do them all justice. And in addition, I suppose for some of these fields, while I have a passing knowledge, I would by no means claim to be an expert in every aspect of microbiome research, and that's why we have some very good work package leads who are responsible for specific topics within the larger master project. So one of those leads is Angela Sesic, who's based in Austria, and she's leading the Plant and Soil Microbiome Initiative. And the objective here is to look at microbes and employ them to improve nutrient uptake, to provide biocontrol for Fusobacterium and to control mycotoxins, specifically in fodder crops, so maize and grass. And to also develop microbiome-based diagnostic approaches in order to detect pathogens, but also to look at existing microbiomes to try to predict to what extent those microbiomes will be responsive if we do try to introduce new inoculants and new consortia to them. And so for each of these, I'm just going to present 2 or 3 slides in the interest of time. But for this project, there's already been significant developments relating to a number of different biofertilizer strains for fodder maize, a number of others for fodder grasses. Those of you who are experts in these particular fields will recognize some of these particular species. There's a number of field trials that are already ongoing in this regard, And the sort of things that we're looking at are different agronomic traits, looking at the level of colonization of the communities that we introduce, as I noted, trying to control mycotoxin concentrations and ultimately determine the quality of the resultant fodder and silage. And so we've already done some smaller experiments and already have data that we can share now, even though it's somewhat preliminary, which shows that, for example, different bacillus strains, and I'm not including the strain names here just for the interests of intellectual property, but some of them have different impacts on the mycorrhizal colonization, and that impact is to some degree dependent on what's already there, so what concentration of AMF are already present in the substrate. I also mentioned diagnostics, and so with the help of QIAGEN, we're working towards the development of kits that will enrich microbial DNA from plant material, because as many of you will know, when you do shotgun sequencing, the other material, the host material, the human or the animal or the plant DNA is also present and could potentially saturate your output from your shotgun data. And so the enrichment of microbial DNA is particularly important there. To try to improve available sequencing pipelines for the detection of relevant pathogens, to identify microbial markers that will, as I mentioned, indicate the responsiveness of a particular community to specific inoculants. So it may be that you need to have a particular type of community for one type of inoculant, and a different community would suggest then that maybe a different inoculant is required in that particular scenario. In all cases, and this is true across all of the different areas, there will be a need for further development and validation of sequencing and analysis pipelines. And obviously all of this involves very many different microplot trials and ultimately larger field trials at the end of towards the end of the master project. So I'll move on to marine microbiomes, and the marine microbiomes area is being led by Viggo Martinsson at Mattis in Iceland, with a number of other partners involved as well. So what their objective here is to develop microbiome-based solutions to increase bioavailability of sustainable feeds for enhanced fish growth, and also to employ microbiome-based diagnostics. So it's a similar theme to the plants and soils, trying to introduce strains that will improve things and also to better detect undesirable strains. And I should have noticed previously that one of our partners is Oxford Nanopore, and you may already be aware that they're producers of handheld DNA sequencing devices, most notably the MinION. And having them as a partner makes a lot of sense, in that while there are other technologies such as Illumina that we will be using during the course of this project, it's not practical for farmers and food producers to all have access to Illumina high output type instruments. And so it's important to be able to develop handheld devices such as the MinION that will be available to people to do real-time sequencing in their own facilities, send their data to the cloud, and then get very rapid, high quality data back. So what Viggo and his colleagues are doing are to carry out comparative growth trials with juvenile salmonids that are fed different types of proteins and also some prebiotics. So these are nutrients that are used by gut microbes, by desirable gut microbes to grow, and probiotics, which are health promoting microbes themselves, and then to also employ pathogen detection approaches using this Oxford Nanopore or ONT sequencing technology. And so again, we already have some nice data coming through relating to being able to compare the impact of the introduction of different meals and different forms of single-cell protein and prebiotics on growth, and also in turn to detect the impact on the gut microbiome as a consequence of this. With respect to pathogen detection, you can see this aerial shot of different locations relative to aquaculture cages. So this is fish farming, and to determine where the pathogens are located, right next to the cages and further afield. And one can do 16S sequencing with Illumina, but that will just get you down to genus level assignment, and it's hard to determine if there really is a pathogen of concern there. Whereas by doing nanopore shotgun metagenomic sequencing, it was possible to detect a number of different fish pathogens, and then to introduce particular approaches to try to address those and control them going forward. The Rumen microbiome area is so large that I could have spent an entire hour just talking about that alone, and so it's going to be difficult to do it justice in 2 or 3 slides, but I'll do my best. So what's the objectives here, and this is being led by Sharon Hughes at Queen's University in Belfast and David in CSIC in Spain. And so they're utilizing host genomics and studying host genomics to assess their impact on rumen microbiome and feed conversion efficiency, and animal phenotype in terms of milk and meat quality. They want to define the rumen microbiome at different times of life in order to, again, improve efficiency, reduce greenhouse gas levels, and also to develop different microbiome-based tools to predict an animal phenotype, in that the rumen will give you a lot of information, but it's not always possible to very easily get access to a rumen sample. So instead, if you were to collect a buccal sample from the mouth of a cow or a goat or a sheep, can you determine its microbiome on the back of that and then determine what feeds they should be consuming? And so we're fortunate that we have very powerful infrastructure within the consortium that can, and again, I'm just using one example, relates to methane production and quantifying it. So these are green feed approaches whereby, as you can see here, it's an infrastructure whereby the animal inserts its head, and while feeding, it will also release methane. Methane's released in the form of, I guess, burping of cows, and so you can determine to what level methane is produced. And you have similar portable accumulation chambers which are used for sheep. And this is the setup of the arrangement that we have in Spain for looking at goats and to determine their feed efficiencies and various other different parameters as well. We also have access through our partners to different systems that allow us to determine milk quality and to detect what levels of healthy fats are present, looking at different sensory profiles and different pathogen levels and so on and so forth as well. And again, this is just one of 20 different examples that I could have taken, whereby in order to look at host genomics, we have the very powerful opportunity to look at identical twins. And this is through our partners Luke in Finland, who look at who are able to separate twins into separate groups and to compare and contrast the impact of a different intervention, safe in the knowledge that you're comparing two animals that have the same genotype, and therefore determining to what extent the intervention versus the genotype is dictating what microbiome is present. We also have a large focus on the microbiomes of food processing, waste streams and gut health, as you might have gathered previously, and this is led by Elke Jorent in University College Cork in Ireland and Danilo Ercolini in Naples, along with these various other different partners as well. I'll speak in relation to byproducts initially, and lots of people will be familiar with whey as a byproduct of the cheese industry, where in previous decades, whey was an inconvenience, and producers of cheese were trying to figure out what to do with it in terms of putting it in landfill sites or what to do with that waste. And then ironically, whey is the more valuable component of cheese production, in that it's in great demand for initially bodybuilders and then athletes, and now the general community are conscious of the benefits of whey in terms of putting on muscle mass and avoiding sarcopenia and whatnot. And lots of other industries are conscious of this potential for their waste streams as well. And that's true in brewing also, where there are a number of different waste streams relating to rootlets, or brewer spent grain, or brewer spent yeast, and other materials that in the past would have been waste, but contain rich sources of microbes and protein and other materials that can be used itself, and fiber that can be used directly themselves, or that can also be fermented by microbes to increase the bioavailability of the nutrients, and also to remove any toxins or off flavors or undesirable components. And so we've already created some products and different pastas and breads whereby we've tried to and succeeded in increasing the fiber levels in these various different products as a consequence of using the brewer spent grains from the brewing industry quite successfully. And these, because we're working with Anheuser, it means that this is not just theoretical, but they're now beginning to put this into practice in terms of the materials that they're generating from their products. This slide you might recall earlier, where I showed you how we looked at the food chain for the production of milk powders. But we're now extending that to look at lots of different food processing environments. So rather than just looking at a small number, we're doing this right across Europe and have begun sampling across 200 different companies from multiple different companies in different areas of of food production. And by doing that, as I say, we can get a better determination of just what are the microbes that are present in the community. We can improve databases, but we're also developing standard operating procedures that are not relevant just purely to scientists, but are so straightforward that a non-expert, a non-microbiologist in a food production plant can employ these technologies in the future. And so before COVID, we had made substantial progress, and we're beginning to ramp up again now. So we're over maybe almost 2/3 of the way through in terms of the number of companies that have been visited. And these companies, a lot of them are dairy, just reflecting the research interests of the various different partners, but we also have a number of plants that produce meats, vegetables, seafood, ice cream, and others as well. So we're going to really get a very large amount of data, hundreds of samples from each individual plant, ultimately coming up with thousands, tens of thousands of different samples, and ultimately the corresponding data to go with it. And so I'm going to tell you about some other work that we're doing in parallel that's been done previously, but that has also fed into our work within Master. And this relates to our work relating to studying fermented foods from lots of different environments. And not just the traditional fermented foods, but foods from right around the world, and some that you might be less familiar with. And so by looking at all of these different weird and unusual fermented foods, we were able to see what microbes are present there as well and how they may contribute to health. We're seeing that the microbial communities differ. Again, this is a PCA. Each dot represents a different microbial community, and how closely located they are represents how similar or dissimilar they are. And you can see the microbial communities from dairy products are somewhat related. Those from brine-based foods, sauerkraut and kimchi, are also more related. And those from sugar-based fermented foods, so these, for example, would be kombucha or water kefir, are also somewhat similar as well. And through this sort of approach, you can identify a number of completely novel species, and this was done by John Leach from our team. And we can look to see what other attributes and health-promoting genes are encoded with all of these different foods. Now that's of relevance because within Master and in Danilo Ercolini's team, we have been able to take advantage of this data. So we've coupled John's previous data relating to fermented foods with some more data that's been generated by Aaron Walsh when he was part of our team together with Garino McCory. And in that instance, what we did is carried out the largest survey of cheese microbiomes around that's been carried out to date. And this involved a large number of producers in Ireland, but also combined with previous data from around the world. And this paper is currently under review. It's close to being accepted, we hope, and so should create a lot of attention, I think, in the coming weeks and months. And from this, we can see all sorts of different things relating to which are the most dominant microbes that are present, what's the different relationship between viruses and yeast and fungal populations relative to bacteria. You're seeing here the relatedness between particular microbes that are present in the cheese and viruses, and how, depending on the ratio present, that relationship can be positive or negative. But we can also look at this to find CRISPRs and antimicrobial resistance genes and flavor encoding genes and lots of other things. So again, this is the sort of study that could take an hour to really do it justice on its own, but I won't linger on it any longer. But suffice it to say, what we've done with Danilo is to combine our different fermented food microbiome data and compare that with lots of gut microbiome data to see to what extent the two overlap. And so we've done that for lots of different lactic acid bacteria, which you find both in the gut and in fermented foods, and to see to what extent they overlap. And so from this tree, you can see, using Streptococcus thermophilus as an example, a number of different strains or strain groups that are present both in gut samples, which are indicated in the orange colors and the inner part of the circle, and also in blue. And then there are other blues that represent strains that are just found in foods, but not so much in the gut. So you're seeing the net outcome, I guess, from the study is to show just how great the contribution of fermented foods is to the levels of lactic acid bacteria in the gut, and the identification of fermented foods as a source of potential health-promoting microbes. And then I'm conscious of time, so I'm just going to do a very quick whistle-stop tour of the last area, which relates to our employing new integrated microbiome technologies, led by Nicola Sagata in Trento and Chris Creevy in Queens. And so what we're doing there is they're helping all of the other parts of Master by providing bioinformatic assistance. They're developing new tools that will be more easily employed by industry, that will be user-friendly, and then also performing some meta-analysis. And so we're developing different standard operating procedures for these companies, developing pipelines and tools that will be available for the researchers, but also resources that will be made available to all that will, for years after Master ended, can be a source of food chain microbes that will be of use when assembling things and identifying, assigning taxa and various other different things. Finally, I'm going to give thanks to our advisory boards, our scientific and industry boards, and again to name check Kelly and to thank her for her contribution in that regard. We really appreciate their contribution. I will, before acknowledging others, I'll make a quick pitch with relation to World Microbiome Day, which is on the 27th of this month, so a little over a week away. And so I would encourage you all to get involved in that initiative. There's a specific website, there's lots of activity on Twitter and on Instagram. And so we would encourage anybody doing microbiome research around the world to get involved. It's being led by Microbiome Support and NAPC Microbiome Ireland. We want you to let us know what you're doing, how you're going to contribute, and we really want to take advantage of this to make all of the globe aware of just what's happening in microbiome research and how it can contribute to have a beneficial effect on their everyday lives. It would take too long to acknowledge and to thank all of the very many different people in the Master Consortium, so I won't even try, but thank you. I will make one specific reference to Mairead Coakley, who's our project manager, without whom this could not be a success and everything would have failed long ago. So thanks, Mairead, for keeping the show on the road. These are the various different specific collaborators within my team or with my team who allowed us to get to the point where applying for Master and putting Master together was practical, and who also provided some of the additional data that I presented to you here today. And thanks to all of the funding agencies, and a final thank you to all of you who connected in today and took the time to listen. Thanks very much.
Great, thank you very much, Paul, for an excellent overview of a very complex project, and I'm sure it becomes challenging just to manage that. We've had several questions coming in, and I'd just like to remind everyone to please use the Q&A for asking questions. And if we'll try to get to them today. If we do not get to them, you will receive answers subsequently directly from Paul or from one of the other organizers. So we've had several questions related to the technology, and you mentioned the partnership with Oxford Nanopore. Are you planning to use any other technologies, any other sequencing platforms in particular, perhaps the PacBio Hi-Fi Sequel 2?
Yeah, so what we're going to focus on Illumina and ONT sequencing. Illumina, just because it's the industry standard, and I think 90%, over 90% of all sequence data is generated by Illumina. And in very many cases, in order to generate good databases, it's first necessary to really employ a sledgehammer approach and get as much sequence data as possible. And so we'll be using NovaSeq sequencing for that particular purpose. For the long-read technology, I guess what we wanted to do is focus on a technology that was as convenient as possible in terms of being handheld and being devices that we could bring into the field or into a food production facility, and hence the particular focus on ONT. So it's not any particular bias against PacBio, and we've used PacBio in other projects previously, but in this particular instance, I think the need for rapid turnaround and to make this as translatable to industry as possible meant that for the long-read technology, that's why we spoke with ONT initially.
Great. Just looking for one of the other questions. Regarding the plant and soil microbiomes, Is it that the ultimate goal is to introduce inoculant and manipulate the soil microbiome for better plant growth, or is it by using optimization or testing of different management practices to see how they influence the soil microbiome?
There's probably more of a focus on the introduction of strains and inoculants in communities rather than on practices. There is an aspect of practices, but the challenge was, again, we could have dedicated our entire project and all of our budget just to the very many different things that one could do in the plant and soil space. I'm conscious that one or a number of microbes in the overall volume and number of microbes that are present in the soil community, it's challenging. So it's not necessarily always with a view to having a change on the entire microbiome in the environment, but rather having an impact that can still be successful despite the relatively small percentage of microbes that we're adding. And that can be due to a specific direct impact on the host, on the plant, or through the removal of a specific undesirable microbe, through the production of an antimicrobial or some other compound that's having a negative impact on a disease-causing microbe. Sometimes it's just simply competition for space. So by introducing a bacillus or a particular other microbe, then they're occupying a niche and therefore making it inaccessible to a pathogen that's entering. But to answer your question more succinctly, there is more of a focus on the strains than on practices, but there is some component of investigation of practices as well.
Great, thank you. One of the other questions that came in was that the understanding is that essentially the goal of this is to improve all microbiome environments that were mentioned to benefit agriculture as a whole. Is that correct? So it's really looking at the entire chain.
Yes, yeah. So it's from soil to society, or from is one of the terms that's coined, or from farm to fork. So it really is right throughout the food chain, and that was one of the challenging components when we initially saw the suggested area, the suggested topic that the EU put out. But I think in hindsight, it's worked out well in that a lot of the tools and the challenges that we face within individual niches are relevant right across the whole chain. But it's quite difficult without having a large project and a large amount of funding to bring all of these different groups together. And we would typically work in silos and be somewhat unaware of what our colleagues in these different areas were doing. And so it's really only by this joined up thinking that we can really put all of this together. So for example, by virtue of focusing on forage crops and grass, we can link those into the Rumen work package. Some of the waste streams from the spent grains. So that waste stream can be valorized and have value added to them, and then also be used as materials that can be used as foods or feeds, and linking into other work packages. And so I think the value of having this large team and the different expertise together and all talking to one another means that the sum of the parts is much more than all of us individually.
Great. So there's a couple of questions regarding expansion into other areas such as poultry. Do you have any intentions to do that?
So I think not specifically within Master, but I think the lessons that we learn from Master are attributable to other niches as well, and I'm sure we learn lessons as to best practice from an SOP perspective. So the SOPs, for example, that are being developed for monitoring processing environments can also be used for production environments. So which swabs should you use? Which DNA extraction kits should you use? What's the best? What concentration of DNA you should have for MinION sequencing versus GridION sequencing versus NovaSeq. So I think all of those different SOPs will be of value. And I didn't get into it during my presentation, but our project is one of 4 different projects that was funded by the EU in this particular microbiome space. And some of those other projects do have an interest in the poultry space, and they're doing some research in that area. So they're out of the whole the broader EU funding in this area, there will be some poultry data being generated as well.
Great. So just to follow on a little bit with regards to that, are there ways in which projects that are outside of the EU that are focused on some of these other areas can become engaged or participate in some way with your project or try to collaborate with your project?
Yeah, I don't see why not. So it's just a matter of working out the specific way in which to interact. But I'd encourage people to contact me and we can discuss that. So, I mean, ultimately, the goal is to develop new approaches and products and strains and so on that will be made available for society and to benefit society. So by forming links with other like-minded researchers and consortia, I think would be increasing our critical mass to an even greater extent. And so it makes sense to form partnerships. And so I think that would be a sensible approach. And so I would encourage people to contact us with a view to teasing out those options.
All right. We have several questions regarding the rumen work. One is the animals, all of the animals that would be involved in that, which species. And the other one is much more specific, related to the use of twin data. How much of the challenge is there related to data gathered from a variety of host genomes or even the animal's mother behavior? And what strategies are used to negate these?
Yeah, so it's as I alluded to earlier, the Rumen work package is enormous, and so we have lots of different activity across multiple different space there. So I only specifically refer to the cows, goats, and sheep, but within each of those, there are multiple different trials involving different animal, different particular strains and breeds of animals, and so we'll see lots of fluctuations and variations there. With regards to the genotyping and other impacts, we're fortunate to have some contributors from INRA in France who are very much focused on the statistics and data processing side of all of this and making sense of and modeling all of this. And so we're conscious that in each instance that these studies have had to be specifically powered in a manner that ensures that it can be done in a meaningful way. But I've really been excited by the way in which this research has proceeded, because the Rumen team that joined the larger master consortium were a group of researchers who already had a track record of working together on other projects such as Rumen Predict and a number of other EU projects. So they came already ready to go and with lots of exciting data that they've been able to leverage on. And so without getting into great specifics, just because I probably wouldn't do it justice and it's not my specific area of expertise, it is something, those are questions that we're particularly conscious of and have plans in place to ensure that we can get data that's meaningful and that it won't there won't be so much noise that we won't ultimately be able to see the wood from the trees.
Great. We have time for one more question. You mentioned the term risky microbiome several times. How do you define that? And have you ever found a harmless one?
A risky microbiome. Yeah. So what I mean by a risky microbiome depends on the specific context. And so I mentioned it at the start relating to the microbiome, for example, in Nunu. And so in that instance, you're relying on a fermented food and the fermentation process to remove food spoilage and food pathogenic microbes. And so if the fermenting microbes don't become established or aren't present in a sufficiently high number, you're not getting a successful fermentation. And so you have microbes that are potentially disease causing. And so it can be difficult for some communities to determine what exactly constitutes a healthy or an unhealthy microbiome. And so that's still being teased out in some scenarios, but in others it's very clear where you have disease causing microbes, spoilage microbes. So I think by virtue of the large amount of data that we're going to be generating through master, we will have a better sense as to what really is normal and abnormal across these different environments, and then be able to introduce interventions in order to direct that community back towards a desirable scenario.
Great. Well, thank you very much for your time today, Paul. This is a really exciting project, and I have to say I'm excited to be on the advisory board, as I think it's going to be really stellar to see the going really from the fundamental science all the way to translation. I think that's really great. So this concludes our webinar today, and I just will remind you that we have another one coming up in July. We are always looking for ways to present the latest science and science projects. So if you have any ideas about webinars, be sure and reach out to me, and we'd be happy to try to pursue those. So thank you for joining us, and we will see you next time. Bye-bye.